Hello students, today we will learn about cultured cells, that is biology and characterization of cultured cells. So let's begin. It is an accepted fact that in vitro cultured cells do not possess the same characteristics as the in vivo cells. This is mostly due to the changes in the cellular environment. Now let's know about the characteristics of cultured cells. Some of the important distinguishing properties of cultured cells. Now we will discuss. First point is cells which do not normally proliferate in vivo can be grown and proliferated in cultures. Second point is cell to cell interactions in the cultured cells are very much low. Third point is the three-dimensional architecture of the in vivo cells is not found in cultured cells. The normal the hormonal and nutritional influence on the cultured cells differs from that on the in vivo cells. Frequently, cultured cells cannot perform differentiated and specialized functions. Sixth point is the environment of the cultured cells favors proliferation and spreading of unspecialized cells. Now let's know about environmental influence on cultured cells. The environmental factors strongly influence the cells in culture. The major route through which environmental influence occurs. Now we will discuss. First point is the nature of substrate or phase in which cells grow. For monolayer cultures, the substrate is a solid, example plastic, while for suspension cultures it is a liquid. The composition of the medium used for culture, nutrients and physiochemical properties. Addition of hormones and growth factors. And next point is the composition of the gas phase. Next point is the temperature of culture incubation. The biological and other aspects of cultured cells with special reference we will now discuss. First point is cell adhesion, cell proliferation, cell differentiation, metabolism of cultured cells, initiation of cell culture. Sixth point is evolution and development of cell lines. Next point is let's know about cell adhesion. Most of the cells obtained from solid tissues grow as adherent monolayers in cultures. The cells derived from tissue aggregation or subculture attach to the substrate and then start proliferating. In the early days of culture techniques, slightly negatively charged glasses were used as substrates. In recent years, plastics such as polystyrene, after treatment with electric ion discharge, are in use. The cell adhesion occurs through cell surface receptor for the molecules in the extracellular matrix. It appears that the cells secrete matrix proteins which spread on the substrate. Then the cells bind to matrix through receptors. It is a common observation that the substrates with previous cell culture are conditioned to provide better surface area for adhesion. Cell adhesion molecules. Let's know about cell adhesion molecules. Three groups of proteins collectively referred to as cell adhesion molecule CAMS are involved in the cell-cell adhesion and cell substrate adhesion. Now let's know about cell-cell adhesion molecules. These proteins are primarily involved in cell-to-cell interaction between the homologous cells. CAMS are of two types, calcium-dependent ones, that is cadherins, and calcium-independent, that is CAMS. Let's know about integrins. These molecules mediate the cell substrate interactions. Integrins possess receptors for matrix molecules such as fibronectin and collagen. Proteoglycans, let's know about the proteoglycans. These are low affinity transmembrane receptors. Proteoglycans can bind to matrix collagen and growth factors. Cell adhesion molecules are attached to the cytoskeletons of the cultured cells. Now let's know about cell proliferation. Proliferation of cultured cells occur through the cell cycle which has four distinct phases. First is M phase. M phase, G1 phase, S phase and G2 phase. Let's know about M phase. In this phase, M means mitosis. In mitosis phase, the two chromatids which constitute the chromosome segregate to daughter cells. Now, let's know about second phase, that is G1 phase. That means this is the gap 1 phase. Gap phase, this gap 1 phase is highly susceptible to various control process that determine whether cells should process towards DNA synthesis, re-enter the cycle to take the course towards differentiation. Now let's know about S phase. This phase is characterized by DNA synthesis wherein DNA replication occurs. Now let's know about last phase, that is G2 phase. This is gap 2 phase that prepares the cell for re-entry into mitosis. The integrity of the DNA, its repair or entry into apoptosis. If repair is not possible, is determined by two checkpoints at the beginning of DNA synthesis and in G2 phase. Now let's know about control of cell proliferation. For the cells in culture, the environmental signals regulate the cell cycle and thereby the cell proliferation. Low density of the cells in a medium coupled with the presence certain growth factors, example epidermal growth factor, platelet derived growth factor, allows the cells to enter the cell cycle. On the other hand, high cell density and crowding of cells inhibits the cell cycle and thereby proliferation. Besides the influence of the environmental factors, certain intracellular factors also regulate the cell cycle. For instance, cell cyclins promote while P53 and RB gene products inhibit cell cycle. Now let's know about cell differentiation. The various cell culture conditions favor maximum cell proliferation and propagation of cell lines. Among the factors that promote cell proliferation, and first point is, let's know about some important points. First is low cell density, second is low calcium ion concentration, third is presence of growth factors. For the process of cell differentiation to occur, the proliferation of cells has to be severely limited or completely abolished. Cell differentiation can be promoted by some factors. Let's know about the factors. First factor is high cell density. Second is high calcium ion concentration. Third is presence of differentiation inducer. Example, hydrocortisone, nor growth factor. As evident from these factors, different and almost opposing conditions are required for cell proliferation, for cell differentiation. Therefore, if cell differentiation is required, two distinct sets of conditions are necessary. First is optimize cell proliferation, second is to optimize cell differentiation. Now, let's know about maintenance of differentiation. It is now recognized that the cells retain their native and original functions for long when their three-dimensional structures are retained. This is possible with organ cultures. However, organ cultures cannot be propagated. In recent years, some workers are trying to create the three-dimensional structures by perfusing monolayer cultures. Further, in vitro cultures in the cells on or in the special matrices, example cellulose, collagen gel, matrix and glycoproteins also result in cells with three-dimensional structures. Now let's know about dedifferentiation. Dedifferentiation refers to the irreversible loss of specialized properties of the cells when they are cultured in vitro. This happens when the differentiated in vitro cells lose their properties. In the in vivo situation, a small group of stem cells give rise to progenitor cells that are capable of producing differentiated cell pool. On the other hand, in the in vitro culture system, progenitor cells are predominantly produced which ongoing proliferating. Very few of the newly formed cells can form differentiated cells. The net result is a blocked differentiation. De-differentiation implies an irreversible loss of specialized properties of the cells. On the other hand, de-adaptation refers to the reintroduction of specialized properties of the cells by creating appropriate conditions. Now let's know about metabolism of cultured cells. The metabolism of mammalian cultured cells with special reference to energy aspect. Now we will discuss. The the cultured cells can use glucose or glutamine as a source of energy. These two compounds also generate important anabolic precursors. As glucose gets degraded by glycolysis, lactose is mainly produced. This is because oxygen is in limited supply in the normal culture condition, that is atmospheric oxygen and a submerged culture, creating an anaerobic situation. Lactate secreted into medium accumulates. Some amount of pyruvate produced in glycolysis get oxidized through Krebs cycle. A small fraction of glucose enters pentose phosphate pathway to supply ribose 5-phosphate and reducing equivalent NADPH for biosynthetic pathways, example synthesis of nucleotides. Now let's know about glutamine. Glutamine is an important source of energy for the cultured cells. By the action of enzyme glutaminase, glutamine undergoes deamination to produce glutamate and ammonium ions. Glutamate on the transaminase or oxidative deamination forms alpha ketoglutarate, which enters the Krebs cycle. Pyruvate predominantly participates in transmination reaction to produce alanine, which is easily excreted into the medium. In the rapidly growing cultured cells, transamination reaction is a dominant route of glutamine metabolism. Deamination of glutamine releases free ammonium ions, which are toxic to the cultured cells, limiting their growth. In recent years, dipeptide glutamyl alanine or glutamyl glycine are being used to minimize the production of ammonia. Further, these dipeptides are more stable in the medium. As already stated, alpha ketoglutarate obtained from glutamine enters the Krebs cycle and gets oxidized to carbon dioxide and water. For proper operation of Krebs cycle, balancing of the intermediates of the cycle is required. Two metabolites of Krebs cycle, namely malate and oxaloacetate, leave the cycle and get converted respectively to pyruvate and phenyl phosphoenol pyruvate. The latter two compounds can re-enter the Krebs cycle in the form of acetyl coenzyme A. Thus, the continuity of Krebs cycle is maintained. Glucose as well as glutamine gets metabolized by the cultured cells to supply energy in the form of ATP. Now let's know about initiation of cell culture. The cell culture can be initiated by the cells derived from the tissue through enzymatic or mechanical treatments. For details on primary culture, the reader must refer these points. Primary culture is selective process. Primary culture is a selective process that finally results in a relatively uniform cell line. The selection occurs by virtue of the capacity of the cells to survive as monolayer cultures or as suspension cultures. Among the cultured cells, some cells can grow and proliferate while some are unable to survive under the culture environment. The cells continue to grow in monolayer cultures till the availability of the substrate is occupied. The term confluence is used when the cultured cells make close contact with one another by fully utilizing the available growth area. For certain cells which are sensitive to growth limitation due to density, the cells stop growing once confluence is reached. However, the transformed cells are insensitive to confluence and continue to overgrow. When the culture becomes confluent, the cells possess some characteristics. Let's know about these characteristics. First is the closest morphological resemblance to the tissue of origin, that is parent tissue. The expression of specialized function of the cells comparable to that of the native cells. Now let's know about evolution and development of the cell lines. First, let's start. The primary culture grown after the first subculture is referred to as cell line. A given cell line may be propagated by further subculturing. As the subcultures are repeated, the most rapidly proliferating cells dominate, while the non-proliferating or slowly proliferating cells will get diluted and consequently disappear. Now let's know about senescence. The genetically determined event of cell divisions for a limited number of times, that is population doublings, followed by their death in a normal tissue, is referred to as senescence. However, germ cells and transformed cells are capable of continuously proliferating. In the in vitro culture, transformed cells can give rise to continuous cell lines. The cumulative cell number in a culture is represented on y-axis on a log log scale, and while the x-axis represents the time in weeks. The time for development of a continuous cell line is variable. For instance, for human diploid fibroblast, the continuous cell line arises at about 14 weeks, while the senescence may occur while 10 to 20 weeks, usually after 30 and 60 cell doublings. Now let's know about development of continuous cell lines. Certain alterations in the culture, collectively referred to as transformation, can give rise to continuous cell lines. Transformation may be spontaneously occurring, chemically or virally induced. Transformation basically involves an alteration in growth characteristics such as loss of contact inhibition, density limitation of growth, and encourage independence. The term immortalization is frequently used for the acquisition of infinite lifespan to cultured cells. Now let's know about genetic variations. The ability of the cells to grow continuously in cell lines represents genetic variation in the cells. Most often the deletion or mutation of the p53 gene is responsible for continuous proliferation of cells. In the normal cells, the normal p53 gene is responsible for the arrest of cell cycle. Most of the continuous cell lines are aneuploid, possessing chromosome number between diploid and tetraploid value. Now let's know about the normal cells and continuous cell lines. A great majority of normal cells are not capable of giving rise to continuous cell lines. For instance, normal human fibroblasts go on proliferating for about 50 generations and then stop dividing. However, they remain viable for about 18 months, and throughout their lifespan fibroblasts remain euploid. Chick fibroblasts also have in a similar fashion. Epidermal cells and lymphoblastoid cells are capable of forming continuous cell lines. Now let's know about the characterization of cultured cells. Characterization of cultured cells or cell lines is important for dissemination of cell lines through cell banks and to establish contents between research laboratories and commercial companies. Now let's know about characterization of cell lines with special reference to some aspects. Let's know about the aspects. First is morphology of cells. Second is species of origin. Third is tissue of origin. Fourth is whether cell line is transformed or not. Fifth point is identification of specific cell lines. Now let's know about morphology of cells. A simple and direct identification of the cultured cells can be done by observing their morphological characteristics. However, the morphology has to be viewed with caution since it is largely dependent on the cultural environment. For instance, the epithelial cells growing at the center are regular polygonal with clearly defined edges, while those growing at the periphery are regular and distended, that is swollen. The composition of the culture medium and the alteration in the substrate also influence the cellular morphology. In a tissue culture laboratory, the terms fibroblastic and epithelial are commonly used to describe the appearance of the cells rather than their origin. Now let's know about fibroblastic cells. For these cells, the length is usually more than twice of their width. Blastic cells are bipolar or multipolar in nature. Now let's know about epithelial cells. These cells are polygonal in nature with regular dimensions and usually grow in monolayers. The term fibroblastoid, fibroblast-like, and epithelioid are in use for the cells that do not possess specific characters to identify as fibroblastic or epithelial cells. Now let's know about species of origin of cells. The identification of the species of the cell lines can be done by first is chromosomal analysis, second is electrophoresis of isoenzymes, third is a combination of both these methods. In recent years, chromosomal identification is being done by employing molecular probes. Now let's know about identification of tissue of origin. The identification of cell lines with regard to tissue of origin is carried out with reference to the to two characteristics. First is the lineage to which the cells belong. Second is the status of the cells, that is stem cells, precursor cells. Now let's know about tissue markers for cell line identification. Some of the important tissues or lineage markers for cell line identification now we will discuss. First point is differentiated products as cell markers. The cultured cells, that is on complete expression, are capable of producing differentiation markers which serve as cell markers for identification. Some examples, let's know about some examples. First is albumin for hepatocytes, second is melanin for melanocytes, third is hemoglobin for erythroid cells, fourth is myosin for muscle cells or tropomyosin. Enzymes as tissue markers. Let's know about the enzymes as tissue markers. The identification of enzymes in cultured cells can be made with reference to some characteristics. Let's now know about the characteristics. First is constitutive enzymes, second is inducible enzymes, third is isoenzymes, like this. Now let's know about the tyrosine aminotransferase. Tyrosine aminotransferase is specific for hepatocytes while tyrosinase is for melanocytes. Creatine kinase MM in serum serves as a marker as muscle cells, while creatine kinase, that is in short BB, is used for the detection of neurons and neuroendocrine cells. Now let's know about filament proteins as tissue markers. The intermediate filament proteins are very widely used as tissue or lineage markers. For example, astrocytes can be detected by glial fibrillary acidic protein, in short GFAP. Muscle cells can be identified by desmin. Epithelial and mesothelial cells by cytokeratin. Now let's know about transformed cells. Transformation is the phenomenon of the change in phenotype due to the acquirement of new genetic material. Transformation is associated with the promotion of genetic instability. The transformed and cultured cells exhibit alterations in many characters with reference to growth rate, moderate mode of growth, longevity, tumorogenicity, specialized product formation. While characterizing cell lines, it is necessary to consider these characters to determine whether the cell line has originated from tumor cells or has undergone transformation in culture. Now let's know about the identification of specific cell lines. There are many approaches in a culture laboratory to identify specific cell lines. First is chromosome analysis, second is DNA detection, third is RNA and protein analysis, fourth is enzyme activities, fifth is antigenic markers. Now let's know about chromosome analysis. The species and sex from which the cell line is derived can be identified by chromosome analysis. Further, it is also possible to distinguish normal and malignant cells by the analysis of chromosomes. It may be noted that the normal cells contain more stable chromosomes. The important techniques employed with regard to chromosome analysis now we will discuss. First is chromosome banding. By this technique, it is possible to identify individual chromosome pairs when there is little morphological difference between them. Chromosome banding can be done by using Giemsa staining. Now, let's know about chromosome count. A direct count of chromosomes can be done per spread between the 50 to 100 spreads. A camera lucida attachment or a closed circuit television may be useful. Now let's know about chromosome karyotyping. In this technique, the chromosomes are sorted into sequences and then pasted onto a sheet. The image can be recorded or scanned from the slide. The chromosome karyotyping is time consuming when compared to chromosome counting. Now let's know about DNA detection. The total quantity of DNA per normal cells is quite constant and is characteristic to the species of origin. Example, normal cell lines from human, chick and hamster fibroblast. However, the DNA content varies in the normal cell lines of mouse and also the cell lines obtained from cancerous cell tissues. Most of the transformed cells are aneuploid and heteroploid. DNA analysis is particularly useful for characterization of such cells. Analysis of DNA can be carried out by DNA hybridization and DNA fingerprinting. Now let's know about DNA hybridization. The popular southern blotting technique can be used to detect unique DNA sequences. Specific molecular probes with radioisotope, fluorescent or luminescent labels can be used for this purpose. The DNA from the desired cell lines is extracted, cut with restriction endonucleases, subjected to electrophoresis, blotted onto nucleocellulose, and then hybridized with a molecular probe or a set of probes. By this approach, specific sequence of DNA in the cell lines can be detected. Now let's know about DNA fingerprinting. There are certain regions in the DNA of a cell that are not transcribed. These regions, referred to as satellite DNA, have no known functions and it is believed that they may provide reservoir for genetic evolution. Satellite DNA regions are considered as regions of hypervariability. These regions can be caught with specific restriction endonucleases and detected by using cDNA probes. By using electrophoresis and autoradiography, the patterns of satellite DNA variations can be detected. Such patterns, referred to as DNA fingerprints, are cell line specific. In recent years, the technique of DNA fingerprinting has become a very popular and a powerful tool to determine the origin of cell lines. Now let's know about RNA and protein analysis. The phenotype characteristics of a cell line can be detected by gene expression, that is identification of RNAs and or proteins. mRNAs can be identified by northern blot technique while proteins can be detected by western blot technique. Now let's know about enzyme activities. Some of the in vivo enzyme activities are lost when the cells are cultured in vitro. For instance, arginase activity of the liver cells is lost within a few days of culturing. However, certain cell lines express specific enzymes that can be employed for express specificity enzymes that can be employed for their detection. Example, tyrosine aminotransferase for hepatocytes, glutamyl synthase activity for astroglia in brain. Now let's know about isoenzymes. The multiple forms of an enzyme catalyzing the same reaction are referred to as isoenzymes or isozymes. Isoenzymes differ in many physical and chemical properties, structure, electrophoretic and immunological properties, that is Km and Vmax values. The isoenzymes can be separated by analytical techniques such as electrophoresis and chromatography, most frequently electrophoresis by employing agarose. Cellulose acetate, starch and polyacrylamide is used. The crude enzyme is applied at one point on the electrophoretic medium. As the isoenzymes migrate, they distribute in different bands which can be detected by staining with suitable chromogen substrates. Isoenzymes are characteristic to the species or tissues. Isoenzymes of the enzymes are commonly used for cell line detection. Example, lactate dehydrogenase, malate dehydrogenase, glucose 6-phosphate dehydrogenase, aspartate aminotransferase, then is peptidase B. Isoenzyme analysis is also useful for the detection of interspecies cross contamination of the cell lines. For instance, contamination of mouse cell lines with hamster cell line can be identified by using peptidase B isoenzymes. Now let's know about antigenic markers. Cell lines can be characterized by detection of antigenic markers through the use of antibodies. The antigenic markers may be located on the cell surface or secreted by the cells into the culture medium. Now let's know about measurement of growth parameters of cultured cells. Information on the growth state of a given culture is required to first is design culture experiments, routine maintenance of culture, measurement of cell proliferation, know the time for subculture, determine the culture response to a particular stimulus or toxin. Some of the commonly used terms in relation to the measurement of growth of cultured cells are explained now. Now, let's know about population doubling time, PDT, the time interval for the cell population to double at the middle of the logarithmic phase. Now let's know about cell cycle time or generation time, the interval from one point in the cell division to the same point in the cell cycle, one division later. Thus, cell cycle time is measured from one point in the cell cycle until the same point is reached again. Now let's know about confluence. It denotes the culture stage wherein all the available substrate is utilized and the cells are in close contact with each other. Now let's know about contact inhibition. Inhibition of cell motility and plasma membrane decoupling when the cells are in complete contact with other adjacent cells. This mostly occurs at confluence state and results in the cessation of the cell proliferation. Now let's know about cell density. The number of cells per ml of the medium, it's a cell density. Now let's know about saturation density. What do you mean by saturation density? The density of the cells, that is cells per meter ml square surface area in the plateau phase. Now let's know about growth cycle of cultured cells. The growth cycle of cultured cells is conventionally represented by three phases: the lag phase, the log phase, that is exponential phase, and the plateau phase. The properties of the cultured cells vary in the phases. Now let's know about lag phase. The lag phase represents a period of adaptation during which the cell forms the cell surface and extracellular matrix lost during trypsinization attaches to the substrate and spreads out. There is an increased synthesis of certain enzymes, example DNA polymerase and structural proteins, preparing the cells for proliferation. The production of specialized products disappears which may not reappear until the cell proliferation cases. The lag phase represents preparative stage of the cells for proliferation, subculture and reseeding. Now let's know about the log phase. The log phase is characterized by an exponential growth of the cells followed by the lag phase. The duration of log phase depends on the cells with reference to seedling density, growth rate and density after proliferation. During the log phase, the cultured cells are in the most uniform and reproducible state with the high viability. This is an ideal time for sampling. The log phase terminates after confluence is reached with an addition of one or more population doublings. The plateau phase. As the cells reach confluence, the growth rate is much reduced and the proliferation of cultured cells almost stop. This this stage represents plateau or stationary phase and is characterized by low motility of cells, reduced ruffling of plasma membrane, cells occupying minimum surface area, contact inhibition, saturation density, depletion of nutrients and growth factors, reduced synthesis of structural proteins, increased formation of specialized products. The majority of normal cultured cells that form monolayers stop growing as they reach confluence. Some of the cells, however, with replenishment of medium, continue to grow after confluence, forming multi-layers of cells. The transformed cultured cells usually reach a higher cell density compared to normal cells in the plateau phase. Now let's know about plating efficiency of cultured cells. Plating efficiency representing cell colony formation at low cell density is a measure used for analyzing cell proliferation and survival. When the cells at low density are cultured in the form of single cell suspensions, they grow as discrete colonies. Plating efficiency is calculated by plating efficiency equals to number of colonies formed divided by number of cells seeded into 100. The term colony efficiency is used when each colony grows from a single cell. Now let's know about seeding efficiency. Seeding efficiency represents the survival of cells at higher density, that is seedling efficiency equals to number of cells recovered divided by number of cells seeded into 100. Now let's know about cell synchronization. Synchronization literally means to make two or more things happen exactly simultaneously. Simultaneously, for instance, two or more watches be synchronized to show exactly the same time. The cells at different stages of the cell cycle in a culture can be synchronized so that the cells will be at the same phase. Cell synchrony is required to study the progression of cells through cell cycle. Several laboratory techniques have been developed to achieve cell synchronization. They are broadly categorized into two groups. First is physical fractionation. for cell separation. Second is chemical blockade for cell separation. Now let's know about cell separation by physical means. Physical fractionation or cell separation technique based on on some characteristics in use. First is cell density, second is cell size, third is affinity of antibodies on cell surface epitopes, fourth is light scatter or fluorescent emission by labeled cells. The two commonly used techniques, namely centrifugal illustration and fluorescence activated by cell separation, now we will discuss. Now let's know about centrifugal illustration. The physical characteristics like cell size and sedimentation velocity are operative in the technique of centrifugal illustration. Central centrifugal illustrator from Beckman is an advanced device for increasing the sedimentation rate so that the yield and resolution of the cells is better. The cell separation is carried out in a specially designed centrifuge and rotor. The cells in the medium are pumped into the separating chamber while the rotor is turning. Due to centrifugal force, the cell will be pushed into the edge as the medium is then pumped through the chamber in such a way that the centripetal flow is equal to the sedimentation rate of cells. Due to differences in the cells size, density and cell surface configuration, the cells tend to sediment at different rates and reach equilibrium at different positions in the chamber. The entire operation in the illustration theater can be viewed through the port as the chamber is illuminated by stroboscopic light. At the equilibrium, the flow rate can be increased and the cells can be pumped out and separated in collecting vessels in different fractions. It is possible to carry out separation of cells in a complete medium so that the cells can be directly cultured after separation. Now let's know about fluorescent activated cell sorting. Fluorescent activated cell sorting is a technique for sorting out the cells based on the differences that can be detected by light scatter or fluorescent emission by pretending DNA, RNA, protein antigens. The procedure involves passing of the single stream of cells through a laser beam so that the scattered light from the cells can be detected and recorded. When the cells are pre-treated with a fluorescent stain, that is example chromomycin A for DNA, the fluorescent emission excited by the laser can be detected. There are two instruments in use based on the principle of fluorescent activated cell sorting. First is flow cytometer. This instrument is capable of sorting out cells in different phases of the cell cycle based on the measurements of a combination of cell cycle, cell size and DNA fluorescent. Now let's know about the second point, that is fluorescent activated cell sorter, in short FACS. In this instrument, the emission signals from the cells are measured and the cells sorted out into collection tubes. Now let's know about comparison between physical methods. For separation of a large number of cells, centrifugal illustrator is preferred. On the other hand, fluorescent activated cell sorting is mostly used to obtain high grade pure fractions of cells from small quantities of cells. Now let's know about cell separation by chemical blockade. The cells can be separated by blocking metabolic reactions. Two types of metabolic blockades are in use: inhibition of DNA synthesis and nutritional deprivation. Now let's know about inhibition of DNA synthesis. During the S phase of the cell cycle, DNA synthesis can be inhibited by using inhibitors such as thymidine, aminopterin, hydroxyurea, and cytosine arabinoside. The effects of these inhibitors are variable. The cell cycle is predominantly blocked in S phase that results in viable cells. Now let's know about nutritional deprivation. Elimination of serum or isoleucine from the culture medium for about 24 hours result in the accumulation of the cells at G1 phase. This effect of nutritional deprivation can be restored by the addition by which time the cell synchrony occurs. Now let's know about some highlights of cell synchronization. First is cell separation by physical methods is the effective than chemical procedure. Second point is chemical blockade is often toxic to the cells. Third point is transformed cells cannot be synchronized by nutritional deprivation. Fourth is a high degree of the cells is synchrony 80% is means high degree means more than 80% can be obtained in the first cycle, and in the second cycle it would be less than 60%. The cell distribution may occur randomly in third cycle. Now let's know about senescence and apoptosis. As cells grow in culture, they become old due to aging, and they cannot proliferate anymore. The end of the proliferative lifespan of cells is referred to as senescence. Cellular senescence, the growth of the cells is due to is usually measured as population doublings. That means PD, in short PDS. The PDS refers to the number of times the cell population doubles in number during the period of culture and is calculated by the formula PD equals to log 10 of log 10 of number of cells harvested minus log 10 of number of cells seeded divided by log 10 to the power 2. The phenomenon of senescence has been mostly studied with human fibroblast cultures. After 30 to 60 populations doubling, the culture is mainly composed of the senescent fibroblast. These senescent fibroblasts are unable to divide in response to mitotic stimuli. It must be noted that the cells do not appear suddenly, but they gradually accumulate and increase in number during the lifespan of the culture. The different parameters used for the measurement of cell growth in cultures now we will discuss. First is direct measure of cell number. Second is determination of DNA by RNA content. Third is estimation of protein or ATP concentration. Now let's know about measurement of senescence. The direct measurement of senescent cells is rather difficult. Some of the indirect measures are loss of metabolic activity. Second is lack of labeled precursor, that is thymidine incorporation into DNA, certain histochemical techniques. Now let's know about senescence associated beta galactosidase activity assay. This occurs an overexpression of the lysosomal enzyme, that is beta galactosidase at senescence. This enzyme elevation is also associated with an increase in the cell size as the cell enters a permanent non-dividing state. The number of senescent cells in a culture can be measured by senescence associated beta galactosidase, that is SA beta, insert SA beta assay. The assay consists of some stages. First stage is wash the cells and fix them with a fixative and wash again. Example paraformaldehyde. Second is add the staining solution, that is X-gal powder in dimethyl formamide dissolved in buffer to the fixed cells and incubate. Third is the senescent cells display a dense blue color which can be counted. Next is apoptosis. The process of programmed cell death is referred to as apoptosis. The cell death may be initiated by a specific stimulus or as a result of several signals received from the external environment. Apoptosis occurs as a result of inherent cellular mechanisms which finally lead to self destruction. The cell activates a series of molecular events that can cause an orderly degradation of the cellular constituent with minimal impact on the neighboring tissues. Now let's know about the reasons for in situ apoptosis. First is for proper development. The formation of fingers and toes of the fetus requires the removal of the tissues between them. This is usually carried out by apoptosis. Second is destruction of cells that pose threat to the integrity of organism. Programmed cell death is needed to destroy and remove the cells that may otherwise damage the organisms. Example, cells with damaged DNA during the course of embryonic development, they are destroyed. They may result in birth defects. Second is cells of the immune system after their appropriate immune function undergo apoptosis. This is needed to prevent autoimmune diseases, example rheumatoid arthritis. Cells infected with viruses are destroyed by apoptosis. Third is cell destruction due to negative signals. There are several negative signals with the cells that promote apoptosis. These include accumulation of free radicals, exposure to UV rays, X-rays and chemotherapeutic drugs. Now let's know about mechanism of apoptosis. The programmed cell death may occur due to three different mechanisms. First is apoptosis due to internal signals. Second is apoptosis triggered by external signals. Example, tumor necrosis factor alpha, TNF alpha, lymphotoxin. Third point is apoptosis triggered by reactive oxygen species. Now let's know about role of cap- caspases in apoptosis. A group of enzymes, namely activated proteases, play a crucial role in the programmed cell death. These proteases are actually cysteine aspartate specific proteinase or in short commonly referred to as caspases. There are about 10 different types of caspases acting on different substrates ultimately leading to cell death. For instance, capsase 1 cleaves interleukin 1 beta. Now let's know about inhibition of caspase activities. Since the caspase are closely involved in apoptosis, it is possible to prevent cell death by inhibiting their activities. Certain specific peptides that can inhibit caspases and thus apoptosis have been identified. Now let's know about measurement of apoptosis. A simple and easy way of detecting head or dying cells is the direct mitosis microscopic observation. The dying cells are surrounded with dense bodies which can be identified under the phase contrast microscope. The cells that have undergone apoptosis contain fragmented chromatin which can be detected by conventional staining techniques. Now let's know about determination of ADP or ADP by ATP ratio. Determination of ADP by ATP ratio. Both the growth and apoptosis of cells require ATP, but when there is growth at rest, an elevation of ADP occurs. Thus, measuring ADP by ATP ratio will throw light on the dead cells. In fact, some assay systems for measuring ADP by ATP ratios are commercially available. Now, let's know about tunnel assay. A significant biochemical event for the apoptosis is the activation of endogenous nuclease activity. This enzyme clips DNA into fragments with free 3 hydroxyl groups. The newly formed small DNA fragments can be extended by employing the enzyme DNA polymerase If labeled nucleotides are used for DNA fragment extension, they can be detected. Tunnel is an abbreviation for TDT mediated DUTP nick end labeling assay. Tunnel is very fast and effective for the determination of DNA fragments formed by endogenous nucleus activity. The apoptic, apoptotic nuclei can be identified by fluorescent technique using fluorescent isothiocyanate, that is in short FITC, and 4,6-diaminophenyl indole. Now let's know about DNA layering test. During the course of apoptosis, the genomic DNA is cleaved to mono and oligonucleosomal DNA fragments. These fragments can be separated by agarose electrophoresis and detected. The nucleosomal fragments of apoptotic cells give a characteristic ladder pattern on electrophoresis. Now let's know about the limitations of the test. DNA laddering test is not very specific since several cells that have undergone apoptosis may not show DNA laddering. Further, some cells not subjected to apoptosis may also show DNA ladders. For this reason, DNA laddering test is coupled with some other test for measurement of apoptosis. Finally finished. Any questions regarding this, you can ask me in the comment box. I will definitely answer the question, and subscribe my channel for to know more updates and to know more biotechnological topics. Thank you.